Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. Okay, in this video tutorial we are going to talk about Pilli and fimbrea. So let me write Pilli and now what are Pilli and what are the fimbrea? Okay, Pilli and fimbrea both are hair-like structure and both are arises from the cell surfaces. Okay, both are hair-like structure. If we see how it will be look like, so let's suppose if this is a bacterial cell. Okay, let's suppose, and what actually happened, we have a, now all of you better know that they have what, they have a flagella, but on the other hand they have what, they have a Pilli as well. Pilli will be long hairs like structure, as you are watching. These are what, these are called Pilli. On the other hand we have another hair like structure or appendages which are arises from the cell surface. We call that what, we call that fimbrea. So fimbrea are shorter, as you are watching. Now these black one representing what, these black one representing fimbrea. Their tips always present in the cytoplasm area. Pilli, if we want to distinguish between the Pilli and fimbrea, so Pilli are actually used in sex process. Okay, now here I will discuss, now let me write, now here I will discuss Pilli, and here and that area I will discuss the fimbrea. Okay, so now let's talk the difference. Pilli are what, Pilli are short, Pilli are longer. What, Pilli are what, Pilli are longer. So let me write, it will be longer. It is longer than what, than fimbrea, and their length will be 9 to 10 nanometer in diameter. Okay, so this will be their length. Now on the other hand, if we see the fimbrea, so fimbrea are what, it is short, S-H-O-R-T, short hair-like structure that arises from the cell surfaces. It is also arises from the cell surfaces, and its diameter will be 3 to 7 nanometer diameter. Their length will be on that way. We can easily see the Pilli on light microscope and on electron microscope as well. So light and electron microscope. But one thing that I must tell you, you can easily watch this Pilli on means light microscope when you stain it. On the other hand, you can't watch this fimbrea on light and electron, you can watch it with what, you can't watch it with light microscope. Due to their small size, you cannot watch it with light microscope, but yes, you can watch it with what, you can watch it with the electron microscope. Okay, so you can easily watch it with electron microscope. The next difference is about their numbering. Fimbrea are almost 1,000 in number. They are 1,000 in number, and they cover whole cells. But on the other hand, if we talked about the Pilli, so Pilli are almost, they are 10 to, their number will be 10. Okay, their number will be 10. Now next difference, if we talked about the next difference, so it is that function. So Pilli are specially used during sexual process. Although there is no true sexual process occur in a bacterial cell, but it is maybe used during sexual process. Like we have a bacteriophage, bacteria means viruses, bacteriophage. If you ask a question from me that how it is used for sexual processes, we have an example of bacteriophage virus. Bacteriophage virus are usually, they start their reproduction cycle, or they start their productions, means they can store their copies inside the bacteria. But on those bacteria which have the 6 pili, what actually happen, the viruses sit on the 6 pili and then they will inserted their genomes inside the bacteria. But it is possible by this 6 pili. So we can say it is used for the sexual processes. Moreover, during conjugation process, when this bacteria want to conjugate, or when this bacteria want to means connected with another bacteria, so then it is possible by this pili. So due to pili, the bacteria will be attached to another bacteria. Means, let's suppose if this is the positive bacteria, so they will attach to the negative bacteria. So during this way, attachment is possible by this pili, and we call that conjugation. During conjugation, what actually happen, a conjugated tube is formed, and if they have the means plasmid, so they will transfer their plasmid through this conjugated tube to another means bacteria. So all this process will be possible by what? By this pili. Okay. And on the other hand, if we talked about the fimbrea, so fimbrea is actually used for the attachment. And other thing is that it is mostly used to attach the bacteria on a solid surface like rocks in a stream and host tissue. Okay, now means rocks in a stream and a host tissue. One thing more, host, let me write host tissue and rocks on stream. More, this fimbrea is also used to create a movement inside a cell, but the movement may be what? The movement may be jerk movement. Okay, so when the cell show a jerk movement, so it may be possible by this fimbrea. Now we have some of the example of the bacteria which have showing the jerk movement due to this fimbrea. The example of such bacteria, we have what? We have the example means E. coli, some strain of the E. coli, and we have another Pseudomonas aeruginosa. Try to remove the spelling mistake by yourself. Now we have another bacteria means Pseudomonas aeruginosa. We have another bacteria, Neisseria gonorrhea. There are one thing more that I forget to mention. These pili and fimbrea are made up of 30 to 20 subunit of the proteins, and these pili and fimbrea are definitely what? They are cylinders. Okay. So I hope you understand about that video lecture. If you like this video, so make sure to hit the like button, share this video, and don't forget to subscribe the channel. Subscribe the channel to get more interesting videos like that. Thank you.